Hello everyone, welcome to We Learn and welcome back to the class Molecular Basis of Inheritances. In the last class of Molecular Basis of Inheritances, we have talked about the structure of the DNA, the three-dimensional structure of the DNA, which was given by Watson and Crick. In today's class, we are basically going to study about the DNA packaging. If we talk about the DNA packaging, the first question comes to our mind is that why a DNA needs packaging? Let's start answering this question. But before answering this question, I have another question for you. The question is, you have to calculate the length of the human DNA. You have to calculate the length of the human DNA if there are 3.3 into 10 to the power 9 base pairs in a human DNA. So if we consider 3.3 into 10 to the power 9 base pair in human DNA, what will be the length? What will be the total length of the human DNA? Please stop the video and calculate the length of the human DNA by your own. I hope you have come to an answer. So let's calculate this. If we talk about the number of base pairs in a haploid cell in human is 3 into 10 to the power 9 base pair. In human or in any organism, the two base pair are separated from each other by a length of 3.4 angstrom. So between the two base pair, the length is 3.4 angstrom. So basically what you have to do is multiply this 3.4 angstrom by 3.3 into 10 to the power 9 base pair. So the answer would be 3.3 into 10 to the power 9 base pair multiplied by length between the two base pair. That means 3.4 angstrom. If you multiply this, the 3.3 into 10 to the power 9 base pair, it would be around, if you solve this, so 3.3 into 3.4 angstrom, that would be roughly equal to 3.3 into 3.4, that would be equal to 11.22 into 10 to the power 9 angstrom. So if we have the length of this much angstrom, if we convert this into meter, it would be around 1.02 meter. And this is the length of the DNA in a haploid cell, because this amount of the DNA is present in a haploid cell. The amount of DNA present in a haploid cell, is what we call C value. In a haploid cell is called as C value. This is very important, so please note down this. So this is very important for your examination. What is C value? C value is the amount of the DNA present in your haploid cell. So if we are calculating the length of the DNA in a haploid cell, it comes out to be 1.02 meter. And it is a DNA content of only single cell. A human body is consists of trillions of cells. So the length of the DNA would be very much. If we consider this size of DNA, that is 1.02 meter DNA, we are going to, this is the meters of the DNA, and we are going to fix this DNA inside a cell of micrometer size. If we talk about the cell size, most of the cell size are in micrometer. So basically we are going to fix this DNA in a size of micrometer, and this is basically present inside nucleus. So the amount of the DNA, the height of the DNA, length of the DNA is around 1.02 meter, and basically you are going to fix into a size of a cell into a size of nucleus. So this DNA, which is pretty much longer, that is around 1.02 meter in a haploid cell, that means 2 point something meter in a diploid cell. Most of the cells of our body are diploid cell. So in diploid cell, the length of the DNA would be around 2 meter, nearly 2 meter. So this 2 meter DNA is to be fixed in a cell of size which ranges in micrometer size. So in order to do this, we have to fold this DNA. Therefore, DNA packaging is one of the most important topic for molecular basis of inheritance. So we know that in order to fix this DNA into the size of micrometer, we have to fold this DNA. So this folding happens with the help of some proteins which are called as histone proteins. So how the DNA gets folded? The DNA gets folded with the help of histone protein. What is histone protein? Histone proteins are basically the low molecular weight. Histone proteins are low molecular weight protein. Histone proteins are basically basic protein. If we talk about DNA, our DNA is negatively charged. Due to presence of the phosphorus atom, our DNA is basically negatively charged. So if we have to fold this DNA, we have to have some bases. That is, we have to have some positively charged amino acid in these histone proteins. So these histone proteins are rich in positively charged amino acid. So positively charged amino acid rich. And positively charged amino acids are basically lysine and arginine. So if we talk about lysine and arginine are basically the positively charged amino acid, the basic amino acid. The nature of these amino acid is basic amino acid. So that these amino acids are very important. And these amino acids are positively charged amino acids. So we have positive charge on histone protein and we have negative charge on DNA protein. That means this DNA can be easily folded around histone protein. So if we have this DNA and we have a histone protein so this DNA can be easily folded because negative charge, this is the positive charge, and because of the opposite nature of the charge, both attract each other. So this DNA gets folded around this positively charged amino acid. So if you have a look, then this DNA is folded around this ball-like structure of histone protein. So this is the histone protein, this is the DNA. Because DNA is negatively charged, it loves positive charge. It have higher affinity. It have high affinity of binding with the histone protein. Therefore, the histone protein, because of its positive nature, the DNA gets turned around the histone proteins. So if we talk about this positive charge, there are 5 types of histone. If we talk about the histone type, there are 5 types of histone. So there are basically the 5 types of histone. These 5 types of histones are H2A, H2B, H3 and H4. These 4 histone proteins are basically present in dimer form. And out of these histone protein, H2A and H2B are basically lysine rich. Whereas these histone protein H3 and H4 are arginine rich. So these proteins are basically the arginine rich basic proteins. So these proteins are basically present in dimer form. So all of these are present in dimer form. And there is a single protein which is known as H1. which is also known as H1. So the fifth histone protein, there are 5 types of histone protein, and this fifth histone protein is basically present in single form, that is H1. And H1 is basically rich in lysine amino acid. So it is rich in lysine. So if we talk about this H2A, H2B, H3 and H4, these proteins are basically present in the form of octamer. So histone are basically present in the form of octamer, and this is the H1. So this is the complete structure. So if we talk about this, so we have this. So this is the H1 and this is the histone octamer. This is the histone octamer. The diagram below basically represents the folding of DNA. So we have this histone protein. We have this histone protein, and this histone protein is basically present in the octamer form. Is basically present in the octamer form, because in each of them, if you have a look around, you will look that H2A is present twice. That means all these units H2A, H2B, H3, H4 are present 2 times, are present in dimer form. That means H2O will be having 2 units, H2B will be having 2 units, H3 will be having 2 units, H4 will be having few units. Similarly, there are number of histone protein, and this is the first histone, this is the second histone. The length of the histone is 10 nanometer, and this DNA is folded around this histone protein. On a single histone, this DNA covers around 1.75 ton. So on each histone protein, DNA makes 1.75 ton of DNA. So there are many histone proteins. This DNA here is basically not covered with histone protein, and this DNA is basically what we call linker DNA, or you can say this DNA is a link between the 2 histone units. So this is a histone, this is another histone unit, and this is the link DNA. So this DNA is basically called linker DNA. So most of the DNA is basically covered around the histone protein. So if we talk about this histone protein here, this is the histone octamer, and on a single histone protein there is 1.75 tons of DNA. So there is 1 full ton and 3/4 second ton. This histone octamer here is basically known as new body, or it is also known as core molecule. So it is also known as core molecule. And there is DNA folded around this histone protein, and 1 ton is complete ton and 1 ton is 3/4 ton. And then there is histone protein, and this histone protein is H1 protein. This histone is basically the H1 histone protein. So this is what we call nucleosome. This is what we call a nucleosome. So if we talk about a nucleosome is this octamer plus this DNA and this H1. So nucleosome is octamer plus DNA plus histone protein. So this nucleosome, octamer and H1 histone protein, and this is very important for your examination, for your NEET examination. So nucleosome is basically consists of octamer and a DNA and H1 histone protein. So this is the basic structure of histone protein. We will continue this DNA packaging in the next session of your class. So thanks for watching the video and see you in the next video.